Hello everyone. Today we will be talking to you about CRISPR. CRISPR is a genetic modification technique that has been around since 1987 and only keeps getting better. So let's begin. CRISPR, short for clustered regularly interspaced short palindromic repeats, has ignited the scientific community due to its ease of use and its widespread applications in genomic alterations in vivo. The CRISPR-Cas9 system is more efficient than any other gene editing method today. Genomic editing involves inserting, removing, or modifying DNA in a genome. With CRISPR-Cas9, multiple genes can be targeted simultaneously. Originally, the CRISPR system was found in E. coli as part of their adaptive immune system from Osaka University researcher Yoshizumi Ishino and his colleagues in 1987. Over the years, scientists have found that CRISPR is an adaptive immune response in some prokaryotes, bacteria, and archaea, which defend against phage and plasmid infection. Applications of CRISPR range in microbiology, agriculture, biomedicine, and animals. In CRISPR is done in 4 major steps and has 2 major components. The 4 major steps include the sgRNA for step 1, and in step 2 you will have the sgRNA and the Cas9 protein. For the third step, the target-specific cleavage will be utilized, and in step 4, the cellular error-prone repair, which knocks out the gene, will be in use. The 2 major components are the Cas9 protein, which is able to cut down DNA like scissors, yes, and the single guide RNA, sgRNA, which is able to recognize the sequence that is to be edited. Now we will go into detail on the 4 major steps on CRISPR. 1. The specific CRISPR RNA is a 17 to 20 nucleotide sequence complementary to the target DNA, and a tracked RNA serves as a binding site for the Cas9 protein. The single guide RNA, sgRNA, contains a sequence that is specific to the target DNA. The tracked RNA, transactivating CRISPR RNA, interacts and binds with the Cas9 protein and helps to maintain the Cas9 protein in an active form. In step 2, you will have the sgRNA and the Cas9 protein. The DNA of interest feeds into the Cas9 complex, and the selected target DNA matches up with a single guide RNA. The PAM sequence, which is short for photospacer adjacent motif, has a 3-dimensional structure, a distinct sequence, and is a 2 to 6 base pair sequence that follows downstream the target DNA sequence cut site. In step 3, you will have the target-specific cleavage. Once the single guide RNA, which is the sgRNA, and the selected targeted DNA are matched up, the Cas9 complex protein targets specific double-stranded DNA cleavage. The Cas9 protein is an endonuclease and has the ability to cut the DNA. Cas9 protein is a microscopic example of scissors that we sometimes use to cut unwanted paper. During step 4, which is the final step, you will have the cellular error-prone repair, which knocks out the gene. In order to stop the chance of error-prone repair by DNA, such as mutations or deletions, the gaps are sealed by enzymes. The enzyme used is the NHEJ non-homologous end joining. NHEJ is referred to as non-homologous because the break ends are directly ligated without the need for a homologous template. This means that the selected DNA is essentially knocked out. So, what does this mean? Now, using CRISPR, scientists are allowed to cut out unwanted DNA, allowing for genetic modification. CRISPR can be used on humans, animals, and study model species if they have an undesirable trait. Some associate the term designer babies with CRISPR since they believe that they can change the DNA of babies. For scientists, this essentially means we can target any specific DNA of interest, such as bad unwanted genes, and cut them out by using the CRISPR-Cas9 method system. CRISPR-Cas9 in locusts successful establishment of an olfactory deficiency line by targeting the mutagenesis of an odorant receptor co-receptor ORCO. Locusts are agricultural pests worldwide and used as study models for entomology. However, the absence of targeted gene manipulation systems for locusts has restricted their applications for research. In this experiment, scientists claimed successful use of the CRISPR-Cas9 system to induce a targeted heritable mutagenesis of the migratory locust, Locusta migratoria. The target sequence of sgRNA was designed to disrupt the gene encoding the odorant receptor ORCO gene and examine the roles of the odorant receptor pathway in the locust. Microinjection of the mixture of Cas9 mRNA and ORCO sgRNA into the locust eggs resulted in efficient target gene editing at the rate of 71.1 and achieved a germline efficiency of up to 81.1%. By crossing strategy, the scientists successfully established the stable ORCO mutant lines. ORCO mutants showed severely impaired electrophysiological responses to multiple odors. The ORCO mutant locust lost an attraction response to aggregation pheromones under crowded conditions. The locomotor activity and body coloration of the ORCO mutant locust did not significantly differ from those of the two other genotypes. This study provides an easy and effective approach by using the CRISPR-Cas9 system for generating loss-of-function mutants for functional genetic studies of locusts and for managing insect pests. The injection to the eggs caused the olfactory gene in the locust to be knocked out. Since there was no longer get to smell, they stopped being pests.